Hey A&Pers, this is Dr. Jones checking in. This is A&P1, Lecture Test 5, Video 6? 7. I think 7. Maybe 6. No, it's gotta be 7. And we're starting in the middle of the bottom of page 8 with astrocytes. 8 and 9. And then we're covering the neuroglia. All right. Let's talk about astrocytes. Astrocytes function to maintain the blood-brain barrier. That'll be the next question. Create a 3D framework, as you can see in the picture here. They repair damaged neural tissue, guide neuron development, and control the interstitial environment. As you can see, they surround completely. These little things are called pseudopods, which means false feet, sort of feet. And these little pseudopods completely surround the capillaries, which are the smallest blood vessels where the exchange takes place. And as such, they monitor what gets from the blood into the interstitial environment and back. So they monitor the permeability. And this is the blood-brain barrier. It isolates the CNS from general circulation. When I talk about general circulation, I'm talking about the bloodstream. So the reason is that there are hormones in the bloodstream that act as neurotransmitters. So if they were allowed into the general brain, in the synaptic clefts of the neurons, in between the neurons, those hormones would act as neurotransmitters, and they would make it seem like messages had been sent that hadn't. So in order to not allow that to happen, these astrocytes form the blood-brain barrier that isolates those hormones and keeps them in the bloodstream, doesn't allow them into the interstitial environment or in the synaptic clefts between the brain cells. Okay. Page 9, which on my note set looks like this, I think. There you go. Okay, and here's the list of all of them. They used to go in the opposite order, but they've since switched these. So we'll get to oligodendrocytes and microglia. Okay, here we are. No. Click. Okay. Ependymal cells. There we are. Ependymal cells line the central canal, ependymal cells, and the ventricles. So the central canal is the hole that goes down the center of the spinal cord, and the ventricles are fluid-filled spaces in the brain, and they're important in lab. If you haven't gotten to them in lab, please pause and catch up and learn about the ventricles. Okay. The ventricles are chambers in the brain, and we need all the ventricles. Chambers in the brain filled with CSF. Next question, what's CSF? Did I skip CSF? I did. CSF is cerebrospinal fluid, and it is the fluid in and around the brain and spinal cord, which will continue to be relevant for a little while. So, and then what else did we need to know? I feel like I've skipped something. Ependymal cells line the ventricles and the central canal. Assist in producing, circulating, and monitoring cerebrospinal fluid. So there's that. Ependyma is the layer of ependymal cells, ependyma. It's the epithelial lining of the central canal and the ventricles, and it's made up of ependymal cells. So the layer of them has a different term than the name of the cells, slightly. It's pretty close though. Okay, next are, the astrocytes, I forgot to mention, surround each and every, there's my little dog, she's barking again. Oh, there's a bird out there, I can hear it. I don't know if y'all can hear the birds. We get such cool birds out here in Rockport. Everywhere. All different birds, all year long. So fun. Astrocytes. I think I need a nap. The astrocytes surround each and every blood vessel in the brain. Question, are there many blood vessels in the brain? Answer, a few. That's a lot of blood vessels. So what they do is they take the brain, a brain of a deceased person, and they inject the blood vessels with this red latex or plastic. It's called plastination. And then they like dip it in acid and dissolve the brain matter off, and you're left with a great visual of the blood vessels in the brain. There's a lot. And each of these is completely surrounded by astrocyte pseudopods. Just to give you a sense of scale here. Okay, oligodendrocytes. I love oligodendrocytes. They're so much fun to say. Oligodendrocyte. Yeah, that's going to be on the test. And they form the myelin sheath in the CNS. And what is myelin? Myelin, that's how you spell it. There it is by itself. Myelin is the electrical insulation around some axons that speeds up the action potentials, which we will talk about more later. And also you will find myelin and myelinated axons in lab. So another reminder to keep up with lab, please. There's lab 4, the terms are all completely different and they take longer to memorize because they are less familiar. So I don't know if y'all have been successful in your attempts to cram lab material, but it won't work for lab 4. I doubt if it worked for the other 3, but it's going to work less for lab 4. If you wait till after the test to study lab, if you wait till the week of the lab test to study lab, you're going to be screwed. Please don't do that to yourselves. Lab takes the information, takes time to process into your brain to the point where you can regurgitate it on demand, correctly spelled in like a minute. Okay, so do yourself a favor and study this stuff like your grade depends on it. Yeah, it's my recommendation. Study it like your grade depends on it. Okay. Myelin, we did that. Myelin sheath is the myelin covering on the axon. So the layer of myelin is the myelin sheath. The myelinated axons are axons with myelin sheath that are completely surrounded by the myelin. Some axons are not completely surrounded by the myelin and they will be unmyelinated axons. Internodes are the myelinated parts, and here you can see where it's covered with myelin. It's an internode. And then the nodes of Ranvier, so you can see right here, node of Ranvier, are the parts in between the internodes. Yeah, I know, it's awkward, right? Like a double negative. So they're the gaps in the myelin sheath where there's no myelin covering. In between 2 myelinated bits is a node of Ranvier. And the IER makes it French, and it's A. I don't know how to make the rules. And no, I don't speak any French other than like a croissant. That's it. So Chevrolet, there we go. Unmyelinated axons are axons that are not completely myelinated, which we will have a picture of here in a second. And the gray matter are the cell bodies, dendrites, and unmyelinated axons. The white matter are the myelinated axons. And to finish off page 9, I think we can get back to doing a page video. Yeah, let's do that. So to finish off page 9, microglia, and it's like micro, like tiny small, glia. Oh, look, it's everything I just said with the picture I promised you. Okay, so here are some unmyelinated axons, and you can see that they're partly covered. And here's a myelinated axon, and you can see that it's completely covered with it wrapped around and around and around and around. It'd be like if you had a breakfast sausage and a thin pancake, like a crepe or a tortilla or something, and you wrapped it around and around and around and around and around. That's the myelin sheath. So say it's a really long breakfast sausage, and each tortilla is an oligodendrocyte, and the layer of is myelin, right? And then the layer of all of the tortillas is the myelin sheath. Okay. And that will be white matter. Mostly because you see how this is mostly cell membrane, so there's like a little bit of cytoplasm out here at the end, but mostly the cytoplasm has been squished out. So there's just layers of cell membrane. And what is cell membrane made of? Fat. So that's why the myelin sheath makes up, the myelinated axons make up the white matter, because they're surrounded by layers and layers and layers of fat. So that's why. Phospholipid bilayer. Microglia. So again, micro means small. The glial cells are the neuron helper cells, but for science we usually emphasize the third from last syllable, so microglia instead of microglia. Microglia. And these are phagocytic janitors. They eat the debris, they eat the waste, they eat any pathogens that come in, and they do wander around, and they're phagocytic, so like amoebas. Remember the amoeboid movement where they surround it and then digest it, and the lysosomes and the, oh, I love them. Okay, so we will call that, because we did like a page and a bit. So we will start with the neuroglia of the peripheral nervous system for the next video. Thank you as always. Let me make sure we got all the pictures, because the last one I skipped a picture. Yeah, that's the next video. Okay. Thank you as always for tuning in. The education must go on. Dr. J out. Study Lab.